Hello everyone. In this video, we are looking at proteins and the large, diverse, amazing group that proteins are. Proteins are very diverse. They range in functions is actually unimaginable. They not only are they used for all the chemical reactions in the body, um, or all the chemical reactions in organic cells, but they are usually used to build a lot of structures. So if we're talking about humans, for example, proteins are super important. They account for more than 50% of the dry mass of most cells. Some proteins actually speed up chemical reactions. You'll learn them as enzymes. And then other protein functions include the defense, storage, transport, cellular communication, movement, structural support. There are a lot of things that proteins do. Let's go over some of those examples. So there are enzymatic proteins. And so these are the proteins that are enzymes. Their function is to selectively accelerate chemical reactions. An example would be digestive enzymes used to break down the bonds in your food molecules. And what they do is they help you digest the foods that are in your body. Defensive proteins. Their function is to protect against disease. An example would be antibodies that are in your blood. Uh, they inactivate and help destroy viruses and bacteria. Storage proteins. So the storage of amino acids, which the storage of energy. Example is casein, the protein in milk. It's a major source of amino acids for baby mammals. Plants have storage proteins in their seeds. Uh, Albumin is the protein in egg white, used as an amino acid source for the developing embryo. These are just a few examples of the many storage proteins that exist. Transport proteins. Their function is to transport things across membranes. An example would be hemoglobin, the iron-containing protein of vertebrate blood. It transports oxygen from the lungs to other parts of the body. There are many transport proteins in the plasma membrane, and we'll cover them at another time. Hormonal proteins. Their function is the coordination of an organism's activities. A great example is insulin, a hormone that's secreted by the pancreas to reduce blood sugar in the blood. Receptor proteins, their function is to um, react and respond to chemical stimuli. For example, the receptors built into the membrane of a nerve cell that can detect signaling molecules released by other nerve cells. Without receptor proteins, we wouldn't be able to react to stimuli um, in our environment. Contractile and motor proteins. Their function is for movement. Examples include the motor proteins that are responsible for the movement of your cilia and flagella. You have cilia linings in your intestines, for example, for the movement of things to go through the digestive system. Your sperm cells have flagella for movement towards the egg. And our muscles are filled with actin and myosin proteins that are responsible for the contraction of our muscle cells. Every movement that your body is making is through the use of contractile and motor proteins. There are structural proteins, which are used for support. An example would be keratin in the hair, horns or feathers, and other skin appendages that are built out of keratin. Insects and spiders use silk fibers to make their webs and cocoons. And then we have collagen and elastin proteins that provide a fibrous network of connective tissue found in our skin. There are lots of proteins, and that was just an examples of several of them, not all of them, and there's a lot that you could study. Enzymes are proteins that act as catalysts to speed up chemical reactions. Enzymes are a group of proteins that have such an important role for chemical um, metabolic function within our cells. Enzymes can perform their functions repeatedly, and they are basically workhorses that carry out the processes of life. And so proteins are constructed from the same set of 20 amino acids. And these amino acids form the monomers of the polymers of proteins. Polypeptides are unbranched polymers built from these amino acids. Okay. The bond between amino acids is called a peptide bond. It's just called, it's just given it's a different name just to recognize that when we're talking about bonding of amino acids, if you can just say a peptide bond. So a protein is a biologically functional molecule that consists of one or more polypeptides. These polypeptides are once again built from the amino acids, which are considered the monomers of the proteins. There are 20 amino acids that are important for biological life. The amino acid structure has an amino group and a carboxyl group. In this image here, you can see this is the amino acid structure with an amino group on one side, carboxyl on the other, a hydrogen in the middle, and then more importantly, an R group. The R group is what can vary and change between the amino acid. It is the R group that helps create the 20 different amino acids that exist. And so here is the amino acids, the 20 of them. You are not required to learn the 20 amino acids, though some of them are more important than others, at least when we're talking about proteins structure. But the 20 amino acids here all have the same basic structure. It's just the R group that is different between all of them. Those amino acids can be bound together to form through peptide bonds, and those are covalent bonds. And then when they are bound together, they form a polypeptide, which is a polymer of amino acids. Uh, these polymers are protein structures. Polypeptides can range in a few monomers to more than 1,000 monomers, and sometimes even larger than that. Each polypeptide has a unique linear sequence of amino acids. This unique sequence of monomers that forms these polymers is what creates all of the proteins that exist. And so this is showing you that to form a polypeptide, it a polymer of protein, you would do it the same way as any other polymer through dehydration synthesis. And so this is showing you two peptide bonds. So these are two amino acids that are bound together, and we want to bond another amino acid to this chain. So dehydration synthesis has to occur. Remember, dehydration synthesis is the loss of water, so it's becoming dehydrated. And so now you have a longer chain of amino acids here. So you have a longer polymer that exists. Proteins vary based on the number of monomers and based on the order of the monomers that are bound. This creates a lot of unique structures. And so it's important to go over structure and function of protein. Remember, structure can directly influence function. So the specific activities of proteins results from their intricate three-dimensional architecture. A functional protein consists of one or more polypeptides um, that are either twisted, folded, or coiled into a unique shape. So learning protein structure is important. The only thing this image is showing you is that these are different ways that um, the book or people in general will identify um, proteins. So these are different protein models that exist. Um, so the sequence of amino acids determines a protein's three-dimensional structure. A protein structure determines how it works. The function of a protein usually depends on its ability to recognize and to bind to some other protein. For example, if you have, for example, something foreign in the body like the flu virus, which has proteins, an antibody will have the similar structure to that protein of the flu virus, and which will allow for it to bind to it, which will allow for it to destroy it. There are four levels of protein structure. The primary structure of a protein is its unique sequence of amino acids. So amino acids will form in a linear chain. The second structure for proteins is that those chains can coil and fold into polypeptides. Tertiary structures are determined by interactions on the various sides of these chains. The coiling and folding of the chains can stay bound together and form tertiary structures. Now, tertiary structures can be grouped together or bound together and form quaternary structures, which results when a protein consists of many multiple polypeptide chains. So we're going to go through each one because that's a lot of information to remember off one slide, but we'll go through each one. The primary structure is a sequence of amino acids. So remember, primary structure for proteins is like just a long chain of amino acids. Primary structures like the order of letters in a long word. The primary structure is determined by the genetic information as it's read. And so later on, when we review transcription and translation, you'll understand how proteins are built. But for right now, let's review primary structure. Remember, primary structure is just a long chain of amino acids. And so as we bind amino acids together, you can see these little circles here represent the different amino acids and the names of the amino acids. They're just bound together. And so this is showing you that there are over 125 amino acids here bound together to form a primary structure, which is just a long chain. Now that long chain will coil and fold and form secondary structures. These secondary structures result from hydrogen bonds between the backbones of the polypeptides of the amino acids. So typical secondary structures are a coil called an alpha helix or a folded structure called a beta pleated sheet. And this is showing you how these look. So in secondary structures, you can have an alpha helix. So the long chain of amino acids start to coil and form alpha helixes, or the long chains will start to fold and form beta pleated sheets. Um, and so these secondary structures are what is used to build tertiary structures, and tertiary structures will bind together to form quaternary structures. Now, a great example of a secondary structure that is important for some organisms, this is a garden spider, but spider silk is made from protein. Spider silk is an excellent example of a beta pleated sheet. So silk that is used to build spider webs is beta pleated sheets. Tertiary structures, the overall shape of a polypeptide, results from the interactions of the R groups. These interactions include hydrogen bonds, ionic bonds, um, more importantly, hydrophobic interactions, and sometimes van der Waal interactions. Basically, what I want you guys to know is that the our groups will interact with each other just like anything chemically will interact with each other. And then sometimes they will bond, like strong covalent bonds called disulfide bridges may reinforce the protein structure. These things will bond together. So in this image, it's showing you that as alpha helixes and beta pleated sheets start to clump together, to bond together, they will form these tertiary structures because they will be bound together. And this is showing you all of the different ways that these structures can bind. You can have hydrogen bonds, you can have disulfide bridges, or you can have ionic bonds. There are several different ways that bonding can take place in tertiary structures. And last but not least, tertiary structures can be put together um, and form a larger macromolecule called a quaternary structure. There are a lot of quaternary structures. Some of them are um, more famous than others. Collagen, for example, is a fibrous protein consisting of three polypeptides that are coiled like a rope. Um, we all know collagen is important because it's what keeps our skin tight and elastic. Hemoglobin is a globular protein that's made from four polypeptides, two alpha and beta subunits. And what I mean by alpha and beta subunits, meaning that it's made from four tertiary structures to make one quaternary structure. So collagen is made from three um, tertiary rope-like structures that are bound together to form a quaternary structure. And then this is an example of hemoglobin, which is made from four subunits, four tertiary structures that are bound together to form one quaternary structure. So those are all the ways that you can build um, proteins. And there are a vast number of them because of that. The ability to get larger and larger um, and to have unique function based on the structure is important. So, what determines a protein structure? In addition to protein structure, primary structure, physical and chemical conditions can affect structure. For example, if you alternate conditions in the environment, such as pH, salt concentration, temperature is a huge one, or any other environmental factor that can cause a protein um, to not be in a normal condition can cause it to unravel. This loss in a protein's native structure is called denaturation. Why is this important? Because sometimes proteins need to be, you'll learn that some proteins in the body are activated and sometimes they need to be turned off. And the way to turn them off means they may need to be denatured to lose their structure. But this is also why some organisms cannot be um, in extreme environments. This is why it's harmful to some organisms. Like for humans, we cannot be in the freezing cold for too long, or we cannot be in the blazing heat for too long because we start to undergo denaturation. A denatured protein is biologically inactive. And so, like for example, extremely high fevers can be fatal because proteins in the blood will denature, and meaning they're no longer active or working. Denaturation can sometimes be reversed. That's not always possible. Actually, for the most part, what we've learned medically is that we cannot allow, we can't rely upon our bodies to fix proteins that are denatured. Usually, the way to fix a protein that's undergoing denaturization is to recycle it. So a lot of chemical processes, when a protein is denatured, it will then get recycled, and then the only way to get that protein back is to rebuild it from the ground up. Now, we have learned to change the shapes of proteins for our benefit. For example, if you've ever done anything to your hair, like get a perm or to get a relaxer, it's all about changing the shape of your keratin proteins. So it's a change in the three-dimensional shape that can change or destroy the function. And so when we get a perm or get a relaxer, we're just changing the shape of our keratin proteins. And what it does is it breaks the R group bonds, and it allows the hair to be stretched or curled, depending on which one we're trying to do. And so then we apply chemicals to help reform the bonds and keep the hair in the same the new structure that we want it to be in. It may not be natural, but we've learned to manipulate the shape of proteins. So it's hard to predict a protein structure from its primary structure. Most proteins go through several stages on their way to being a stable structure. And because proteins are so complex, and a lot of problems can arise from that, and diseases such as Alzheimer's, Parkinson's, mad cow disease, all these are associated with the misfolding of proteins, meaning that there's a problem with the natural protein structure that is forming proteins, either in the brain or within the body. And so, with that, guys, that concludes proteins, at least what we're going to cover now. Later on in another chapter, we're going to review enzymes in depth and review enzyme function. Enzymes are super important, and as a class of proteins, um, they're so important that they deserve their own unit to look at them. In the next video, the last video, we're going to be looking at the nucleic acids.